Good day to everyone. This is the experiment number 10 entitled Effective Hand Washing together with my companion Aladin Vicky Hachon and RCA Wario and me, Janelle Aspin Vickiano, the reporter. Hand washing is the single most effective way to prevent the spread of infections. You can spread certain germs. When we say germs, a general term for microbes like viruses, bacteria, prions, infectives, etc. Casually by touching another person. You can also catch germs when you touch contaminated objects or surfaces, and then you touch your face, mouth, ears, and nose. A proper hand washing technique includes using sufficient amount of soap, rubbing the hands together to create friction, the rinsing under the running water. The use of glove is not a substitution for hand washing. Proper hand washing and habitual cleanliness can also have a good figure to us as a student and a student of a medical course, because according to one of our instruction, small pores in our body is all it needs for a bacteria or other infectives to penetrate our body. So the goal of this experiment is to appreciate the importance of cleanliness and orderliness in avoiding infection and contamination. So our material for this experiment is only consists of two. Number one is the hand soap. We can use bar soaps, liquid soaps, gel, or alcohols if one have, and one plate of agar. For our procedure, first we have to divide the nutrient agar plate into quadrants and label quadrant 1 to quadrant 4. On the first quadrant is your negative control. Do not touch it and attempt not to pass your hands over this quadrant as you complete the exercise. So there's a first quadrant. It can be either top, bottom, or top left, top right. It's up to you. As long as you don't touch it or contaminate it with your fingers or any materials. Touch the quadrant. On the next step is the touch quadrant 2 with two fingertips. On these fingertips, we can have a bare hand without the gloves as a protection, because we have to assess if one hand or the two finger is full of bacteria or any pathogens. On the step number 4, rinse your hand with tap water as you normally would wash. Dry your hands with two fingertips to quadrant 4. On the fourth quadrant, this is the only wash your hands with water without the soap, so we can differentiate the fingers on the quadrant 2 and quadrant 4. On the fifth step, wash your hands with tap water and soap as you normally would. Dry your hands and touch two fingertips to quadrant 3. On the third quadrant, we are able to differentiate the quadrant 4 and quadrant 2 if properly washing your hand does a impact on the spread of the infections, and we have to compare it if or on what quadrant has the most infection. Now we have to incubate the plate at room temperature until the next lab period. So we can use the autoclave or anything that can fasten or hasten the bacterial or growth of our agar propagator. And our last step is to examine the plates for relative abundance of diversity and growth. Or at the last step of our experiment is to differentiate and to see if proper hand washing does an impact on the spread or the mitigation of the bacteria in our hands, and compare it to the other quadrant, the quadrant 1 having no contamination or no touching or crossing of fingers or something. On the quadrant 2 is the finger that has not been rinsed or washed with soap. On the quadrant 3 is the fingers that has been washed with water and soap. And the quadrant 4 is the finger with only washed with water. That's all. Thank you.